How is everyone doing?
Good morning.
Good morning. Guys doing good?
Good morning.
Good morning, David. How are you?
Pretty good.
How are you doing, Professor?
I'm just gonna have a very quick lab today. I'm just gonna go over ELISA's. I'm gonna answer any questions about your tech reports and your problem sets that you might have. And we'll talk about what we will be doing next week. I believe we are doing lab in person next week, right? We're doing the ELISA's next week, from what I remember. I want to make sure. We're supposed to do ELISA's in person. That's what I would like to do. I'm gonna be partially setting them up beforehand. That's why we want to make sure that your problem set question for ELISA is correct and you guys have the correct values, because that's what I'm using to set it up. So we can have a head start on it and we don't have to wait that like extra hour of protein binding. And then you guys will do the rest. It's going to be a little bit like the Weston, where you have a lot of washing and incubation and waiting steps in between. But you'll get to see the final result and then we get to talk about that. And then, yeah, we're gonna go over the schedule in a second and make sure everything is the way I am claiming it to be. Because you guys want to do really good on the second tech report after how you guys did on the first one for sure. So you can catch up. Catch up. I want to make sure what is this tech report too. Okay. So remember that there is the midterm right after spring break. And the tech report is technically due then. What I probably will have you guys do is have the tech report due the following week. Since we are going online for the last module anyway, it's okay if we do things a little bit up down, right? I want you guys to really work on the tech report and get it done correctly. So I'm gonna give you that extra week. So over the spring break, you don't have to worry about it as much and just worry about getting your midterm ready. And then the midterm is online. It's going to be on all the stuff that we've learned so far. And then we will have the tech report due the following week. Okay. But yeah, but we'll talk about all that. Jessica is here. Where's your friend? Where's your sister? Jennifer's not.
She should be
Jessica.
coming soon.
Oh,
I think she's in the other room.
okay, gotcha. So yeah, so the rest of the things, the deadlines will remain the same. I just want to make sure that everyone does really good on the tech report. And we do have time, today and next week to talk about it. So if you guys can do it on time, that's great. But I will give you a little bit of extra time to work on it as well.
Appreciate it.
Okay. Okay, so let's go ahead and get started. Um, First of all, are there any questions from previous sessions before we go forward? Anything on the problem set? Tech report, like I said, I will go over it myself as well, but are there any questions on your part, on your side, that I should keep in mind as I answer them? So I posted a rubric in the tech report to use, and I will post that for each one now. The same exact rubric will be used for the other two tech reports, just so I think it will make it a little bit easier for you to keep yourself on track and to make sure that you're getting all the points that you need to get, right? Because I noticed even though all the instructions that are in the rubric are also there in the written form, sometimes they get lost in translation if they're just like in a paragraph form versus if you have like a little itemized list to do. So I'm gonna see if that helps out. And I use the rubric to grade because it kind of shows you where you're missing out and what things you kind of neglected or forgot about. Um, so that just moving forward, you want to make sure that you're getting everything, all the points in there. A lot of you forgot to either write the narrative text or didn't write it in enough detail, got a lot of points missed because of that. For the discussion, many of you did a great job finding a appropriate resource and incorporating that, but then forgot to deal with like the technical issues and what could have been done better, or why the standard curve would not have looked the way you wanted it to, those type of things. So you want to work on both sides, right? The critical analysis of your own data and then also comparison with published results. So moving forward, that's the other thing that we do want to make sure we incorporate. Share. Some of you did raw data instead of like actual final figures. That last 2 points. So you want to make sure next time no raw data is present in the technical report. Now in the problem set, the more information you give me, the better, right? So if you don't show me the raw absorbances and blank corrected absorbances, there's no way for me to know if you did it correctly or not. And you'll get points taken off because your numbers don't look right. On the other hand, if you give me those numbers, then I can see where the problem is and you can get partial credit. So in problem set, you want to give me as much detail as possible, add the Excel sheet if needed. But in a tech report, you want to give me the final figures and the final information, nothing else. Okay, so just going over the schedule, I just opened it up to make sure everyone's on the same track and knows what's going on. Next week, our lab is in person. We are going to be doing the ELISA, and we are going to see the results from there. It's going to be a fun little exercise, something new. You're going to create a different type of standard curve. So, practice making some dilutions again and doing a lot of washing steps. So we're going to talk about that today. Then the following week, we have spring break, so no classes. Right after spring break, that first week we meet together, we are going to be doing an online midterm through Zoom. So you will have Zoom during class time that you will be working on your midterm on. You'll have your camera on. And you'll work on that stuff online. It is going to be an online exam, so you'll get to do that. Like I said, the tech report is due that week. I will probably push it to the weekend that week or the following week so that you have a little bit of extra time to work on it and make sure that you do it correctly. Now, this was the altered schedule. Having said that, the altered schedule has gotten more altered in that we will not be having any in-person labs after the midterm or after spring break. Because of the way my chemo sessions have worked and because they have to give me a break before they do the next ones, I'm gonna probably be in the hospital many of these weeks. So we're just gonna have Zoom sessions for those, and I will provide you with data from previous semesters to analyze. So that's going to make it, a little bit different, but at the same time, you're still going to be doing the same type of troubleshooting. You're going to be still doing the same type of data analyses. The only thing is you don't get to have that hands-on experience. So you want to make the best of the next week's lab and get some fun hands-on experience in. So yeah, and like I said, we do have that other week in there, which is like kind of a review time too. So we will have built-in times where you can discuss more in-depth data analysis and work on those things as well. So your tech report is going to be due right before the final exam, right? So that's the last thing you're turning in before the final. The final exam is going to be also online. It is going to be take home in a way, right? You don't have to be on the Zoom for it, as it's going to be open probably that weekend all the way through May 3rd. But once you start it, you can't stop, right? So it is going to be timed, and you just need to turn it in on time by noon on Wednesday, May the 3rd. So yep, sounds good. Everyone good so far?
Yep.
Cool.
Yeah.
So that's gonna be our general setup. Now, oops, didn't go where I wanted it to go. That's sad. We'll go back to Canvas. Okay. So this time, right, our module has a lot more things. So remember, as we start thinking about data analysis and tech report, it's going to have a lot more inside it, right? In the results section, you're going to be showing again your standard curve and protein quantification, but then you're also going to be showing your protein quality with that Ponzo stain. You're going to be showing your Weston for Krebs specifically to show how Krebs levels were, generally speaking, between your samples. And then you are going to be quantifying them using ELISA and showing that quantification. But there's going to be a second standard curve with Krebs, and then it's related ELISA. So let's talk about ELISA. There is a PowerPoint and its lecture in the ELISA page that's going to give you more details of that. And I'm actually going to go over some of that. And then once we have the ELISA data, we're going to talk about that next week, not next week, but probably right after spring break, or next week if we are done in time. So let me open this up a little bit because I do want to go over this with you a little bit. So up till now, you have your protein. You've already looked at the Krebs expression. You've already kind of quantified it. You've quantified the total amount of protein, and you've calculated how much protein you're going to need in order to run your ELISA. On the other side, we have plated antibodies for you on top of some 96-well plates that you are going to be using to run your ELISA. Why isn't this showing nicely? I don't like it, but okay, we'll make it. And we are going to be plating your proteins for you as well. I'll probably plate them on Monday so that they have an overnight incubation in the cold, and then you guys, when you come in, you will go ahead and start the rest of the process. Okay, so what exactly is ELISA? ELISA is another kind of an antibody-based assay. It is called enzyme-linked immunosorbent assay. So it's another immunoassay. It detects the presence of antigens, in our case, Krebs, in our biological samples, and relies on antibodies, just like Western's and other immunoassays that we've worked with. So affinity chromatography, where we use the antibody here, Western's where we use the antibody against our specific protein, and this time as well, we're going to do the same thing. So it relies on antibodies to detect whatever target protein or antigen you're looking for, and you want to use an antibody that is super specific to whatever it is that you want to find. So some terminology for this particular type of assay. you obviously need some place for this assay to work in, and that's our solid phase. And our solid phase is typically either a 96 well plate with the high binding properties on the surface of the wells, or it could be strips. So sometimes those 96 well plates come where they're detachable strips or rows in there. So you can just use as many as you need. It protects, the rest of them so you don't waste extras. Then you have to absorb your sample or your antibody to it. So adsorption is the process through which we bind either the antibody or the antigen, so your protein sample or your antibody, diluted in special buffers so that they would attach to that solid phase, that bottom surface of the well. Then obviously the antigen versus antibody still remain the same. Antigen is the molecule that we are going to be looking for, right? So that is the molecule that we are going to be using the antibody against. So antigen in general is any molecule that will elicit the production of antibodies by the body when it's introduced into it. And that's how we create those antibodies. And then the antibodies are the ones that we have produced against that antigen that we're going to be utilizing. So you want to use that pair to get your response. And then it is enzyme linked. And that's because we are going to be using some type of an enzymatic reaction to look at the final output, to look, in either in a colorimetric or a fluorescent or a chemiluminescent way. In that process, the enzyme is going to attach irreversibly to the antibody. Two most common ones used in ELISAs are horseradish peroxidase and alkaline phosphatase, very similar to how we talked about in Westons as well. And then these two can again produce a chemiluminescent signal, can produce a colorimetric signal as well. We are going to be using a chromogen to create a color change so that we can have a colorimetric assay that can be used, that can be examined with our plate reader by looking at the absorbance at the appropriate wavelength. So the chromogen is that chemical that is going to change the color as in response to that enzyme linked reaction that happens when we add the substrate to our system. In many cases, it's the TMB, or similar to what we did last time with the alkaline phosphatase. We could have that reaction as well with the NIPT. Okay, so just going through ELISA, if you imagine this to be one of the wells in your 96 well plate, you are going to have either your antigen or your antibody bound to it. At the very, very basic level, you have your antigen, so your sample bound to the well. You use a primary antibody that is going to be very specific against that protein of interest that you're looking for. It is going to be recognized by it and it's going to bind to it. That primary antibody is then enhanced, the signal from that is enhanced by using a secondary antibody against the animal that the primary antibody was made in, just like ELISA. That secondary antibody is what has the enzyme linked to it, so that when you add the substrate, it's going to react with it, produce a colored product, and that's what you're going to be looking at either in a fluorescent or a colorimetric assay that can be detected and quantified by your plate reader. There are 3 different types of ELISAs that we can look at the most basic level. You can have colorimetric, chemiluminescent, and competitive fluorescent. In a chemiluminescence, you have to use a luminometer to detect the final signal, and that is a light reaction. So you're going to see the production of light. In a colorimetric, you are going to be looking at the color change, and that's going to be examined by the plate reader. The amount of that particular color that is present through the appropriate wavelength. And in a competitive fluorescence, you are going to be looking at the fluorescence that is produced in response to that reaction at the end. There are many ways that we can create the ELISAs. You can have non-competitive or a competitive ELISA. Competitive ELISA is used very little. Most of the time you have one of the non-competitive versions that are created. There are three different ways that you can get non-competitive ELISAs. You can have what we call a direct ELISA, indirect ELISA, and a sandwich ELISA. What you choose is going to be based on the antigen that you're looking for, right? The protein that you're interested in, and its properties, how abundant it is in your sample. Is it very, very abundant, very easy to find, or is it like one of the minor proteins and only like 0.1% of your sample is that? That's going to dictate what exactly you're using, how good the antibodies are against that protein that are currently available. Does it have an ELISA commercially available, or do you have to create one in-house? All of those things are going to dictate what type of ELISA you should be making and you can make. And then what are your capabilities as far as your instrumentation and your supplies are concerned? So we're going to go over each one of these a little bit more in detail. In a direct ELISA, this is the most simple ELISA that you can get. It is great. It is very easy to run. It is, easy to create and easy to use. However, it's only good if your protein of interest or your antigen is really, really, really highly abundant, right? So if it's like lots of your protein, so if we were looking for example, albumin in plasma, so 60% of your plasma is albumin, there's lots of it in there, that's going to be the primary protein present. It's going to be very easy to find. So in something like that, yes, you can run a direct ELISA. The reason for that is it's least sensitive, because in this particular case, the bottom of the well of your ELISA plate is going to be coated with your protein sample. So imagine a situation where, like if, again, you're looking at albumin in plasma, 60%, about 60% of the surface of your well is going to be albumin because that's how abundant it is. But if you're looking at a protein that's only 0.1% of your sample, well, very few molecules are going to make it to your well plate, right? Because there's only 0.1% of your favorite protein in your sample. And most of it is going to be binding of other proteins. So you're not going to have enough sample there to actually detect your protein of interest to get a good signal. So you don't want to use this in situations where your protein of interest is either too small or very little in abundance. The good part is that if you are able to use it, it is again very simple. You can have just a two-step process where you have your protein sample bound to the well. You go ahead and bind it with a specific primary labeled antibody. Again, the antibody has to be very specific, very clean, and has strong binding to your antigen. But if you have something like that, you can have a primary antibody conjugate that already has a label on it, and all you need to do is just detect it. So very easy, two-step process. It is not used very widely because of obvious reasons. It is limited. It's only good for certain low-lying fruits and not for things that are going to be your general proteins that are much less abundant and have a lot of other things that we have to keep in mind. In those cases, we instead use indirect ELISAs of one type or another. And an indirect ELISA, you're going to, again, you can have the antigen, again, your protein sample bound directly to your solid phase. So again, the same thing applies. If your protein is not very abundant, only a percentage of that well is going to be covered with your protein. The rest of it is going to be other things. So keep that in mind. You're wasting a lot of space. Then you are going to utilize, so this is going to be kind of like a Western, where you're going to utilize a primary unlabeled antibody, but it is a very good, specific, strong antibody to your antigen. So you're going to use that to bind to your antigen to amplify that signal a little bit. And then you're going to use a secondary antibody specific to your primary to enhance that signal. And that secondary antibody is what's going to contain the enzyme conjugate that you will then add the substrate for and then look at it in that way. Now, this is going to be obviously a lot more sensitive than direct ELISA because you have that second enhancement step, and it's going to allow for further signal production, and it's going to give you a little bit more kind of flexibility in how much signal you can detect. So if you have a protein that is reasonably sized and it is maybe not the most abundant, but not really super low concentration either, and you have a good antibody pair for it, it's good idea to go ahead and try that, because if it works, that would be an easy step, an easy way to get ELISAs done, quantification done with it. However, if that doesn't work either, and that doesn't work for majority of the proteins also, then you have to do a little bit more more technical ELISA. So antigens like tumor markers, hormones, serum proteins that are not like the main serum proteins, most of the proteins that we are going to be interested in are going to be those, really low abundant proteins. They're in very particular conditions under very particular times, and they're not going to be present all the time. So they're going to be really hard to detect using a simple ELISA like that. In that case, what we do instead is what we call a sandwich ELISA. And that's the type of ELISA that we are going to be running in our lab as well. So in a sandwich ELISA, we use two different primary antibodies against your antigen. We start off by using one of those antibodies. Typically, we use a polyclonal one for this one. Yes, question? I heard something. Maybe it was my imagination. Okay, so for the capture, and we call this the capture antibody. So for our capture antibody, that is going to be coated on the surface of your ELISA plate, on the well of your plate, we use a very specific polyclonal antibody against your protein of interest. What that's going to do is that it's going to create the entire surface of your well as a capture location for your protein of interest, because the entire surface of the plate is now going to bind this capture antibody. Now, because it's a polyclonal antibody, it's going to have lots and lots of epitopes represented all throughout, depending on which antibody molecule you look at. So it's going to give you lots of binding potential against your protein of interest. You then go ahead and put your sample, your protein sample, on top. After you coat them, you can put them away for a couple of weeks, even a couple of months, I think. And then when you're ready to use them, you would add your protein of interest, mixture of interest. So in our case, our brain proteins on top of that well. When we add that sample on there, now it doesn't matter if your protein was only 0.1% of your sample. The only thing that's going to bind to the well plate are your actual molecules, in our case, CREB proteins, because the well is coated with antibodies against CREB. So it's going to saturate your CREB protein and get a lot of them plated. All of them are going to bind. So you're not going to have non-specific binding as much as you did with the others, where if most of it was something else. Here, most of it is going to be CREB. Pretty much all of it is going to be CREB, because you have the antibody bound at the bottom, and the capture antibody is only going to bind CREB. And so you're going to enhance that signal, saturate your protein to get better, more robust signal out of it. Once that is done, you then go ahead and do the rest of it kind of like that indirect ELISA, where you use a second primary antibody against your CREB. Usually the second one that we use is going to be a monoclonal against a specific epitope, away from the others that were there in the capture antibody. And even if you can't, make sure that that's the case. When you use that specific antibody, it gives you a nice clean signal. So we are going to use that second primary antibody. In our case, we're going to actually use the same one as what we use for the Westons. So it's going to bind to it, and then it's going to enhance that signal, right? So now you have a sandwich. You have a primary antibody against CREB, you have your CREB protein, and you have another primary antibody against CREB. That's why it's called the sandwich ELISA. And then you will enhance that signal with the secondary, which has the alkaline phosphatase attached to it. Then we'll add the enzyme conjugate and look at the signal. So you can see how this process will enhance our signal a whole lot more, giving us a lot more robust signal. And we should be able to see the effect, the Krebs amount of Krebs protein very well. And that's going to help us get better quantification. Okay. Any questions on this part? Okay, so then similar to how if you have looked at the Western one too, I've gone through in Westerns, I've given you actual example gels of each type too, but in this one, I've given you some troubleshooting of common problems that you should know about. And what are some reasons that you might see those? You should be aware of them, right? So most of it is going to be involving very little color production or no color production in your positive control samples. So in order to quantify anything accurately, you always have to have a standard curve. In our case, what we're going to be running along with our ELISA's brain samples, right? Our actual brain sample is a standard curve of just pure Krebs dilutions. And so our pure Krebs dilution should definitely give us a signal where the more Krebs is there, the more signal should be there, and you should get some kind of a linear curve. And that curve can then be used to get your equation, and then you can use that to quantify your brain sample Krebs amount. If you have no signal developed in those samples in your positive control, well, obviously the protein, the purified protein could be the issue, but it could also be either that one of your reagent is messed up, or you didn't prepare your dilutions correctly, or your microwell plates were not coated properly with either the Krebs or, in our case, the capture antibody. So we know they are all coated with the capture antibody, so we would be looking at other reasons why it wouldn't work if it doesn't work. If you have samples that have higher amount of Krebs, you definitely see the trend, but it's not very, robust. The more likely situation would be that we are not using the correct method. Maybe we're using direct ELISA instead of sandwich ELISA when that should be the case because your antigen is just not present in a very high quantity. If we can see the color, but we cannot detect it using our plate reader, the most likely reason would be that we did not use the correct wavelength associated with the color change that we are looking at. So you want to make sure that you kind of think through these when you're writing your tech report and talk about those as well. Okay, so let me close this one and then I shall show you the protocol and we will quickly talk about that as well. Questions so far? Yes, no, maybe? I know I say yes, no, maybe a lot. I've noticed that in recordings. But okay, so let's go to the ELISA portion. So like I said, for ELISA, we have already coated our plates for you with the Krebs polyclonal antibody. That's going to be our capture antibody. So when you come in, we are going to be, and on Monday, I will go ahead and do part 1 of the preparation of ELISA plate for you. So I don't know whether I will do that. You can apply 4 degrees. Yeah. So what I will go ahead and do for yours is when you come in next week, you are going to be creating your own standard curve. I will have a master standard curve that I will create. You guys are going to be doing your stuff without that. So, you will have your own standard curve. So you are going to get some Krebs that is pure Krebs, just nothing else in there. It's recombinant Krebs that we bought. And it is going to be at a particular starting concentration I believe the starting concentration is 10 nanogram per 10 microgram per milliliter or 10 nanogram, 10 nanogram per milliliter of our pure Krebs. We are going to use that to create a standard curve ranging from 0 to 5 nanogram per mil. That's going to be our starting sample. You're also going to be using a non-specific protein, which is BSA. So BSA does not contain any Krebs, so you do not expect to see any signal in there. Whatever signal in there you see is kind of like a little bit like a blank where it's the background noise beyond what is present with just no protein alone. Okay, and then you're going to have your own brain sample, not your brain sample, the protein that you extracted from mouse brain, those brain samples that you have diluted to 100 microgram per mil finer protein concentration. So that's the thing you calculated in problem set. For those of you who did not calculate it correctly, I marked it already. So you want to talk to me if you need right afterwards and correct that so that I have your correct numbers. And then you would be plating them into a row of your well plate. Now, there are two ways that we can do it. We can keep it at 4 degrees for, long time. You can keep them in there for an hour at room temperature. The minimum amount of time you would want to keep it at room temperature would be 30 minutes. So we would probably have you guys do this and plate it for 30 minutes. In the meantime, I would have a set of samples already done for you that have already stayed overnight in 4 degrees so that we can process them as well and get some data. Now, once we have plated our samples, allowed them to bind to the antibody. Like I said, at room temperature, 30 minutes is good. We are going to then remove the samples and you are going to wash them, right? All of them with some PBS and the purple PBST to make sure we wash them 3 times to make sure anything that did not bind to our plates gets removed. That's very important because we want to make sure that nothing was left behind, right? That shouldn't have been there, that only Krebs is bound to the samples, and that's the stuff that we look at. We then are going to add our antibody, anti-Krebs antibody to it, and we are going to again incubate it at 37 degrees for 30 minutes. This is our second, this is that, not the capture antibody, but the detection antibody. And we are going to be keeping it at 37 degrees to enhance that speed of our reaction so that they can bind quicker and we can work with them quicker. So we are going to add that in. Once that is added in for 30 minutes, then we are going to do the same steps where we wash them 3 times with PBS tween, and then add the secondary antibody for 30 minutes. Again, we're going to incubate it at 37 degrees to make sure that it works quicker and we are able to do the final detection at the end. To detect the signal, we're going to do the same, the PNPP solution, the AP substrate, and we're going to see the conversion of that product. There's a stop solution that we have to stop the reaction so that it doesn't go beyond the linear range. And then we are going to look at it using the spectrophotometer at 405 nanometer. Okay, so once you have your data, each group is going to subtract the value that they get in A1 from all the other numbers, because that's your blank, that's your background noise. And then you're going to be using wells 1 through 8 to create your standard curve and your equation. And then you're going to compare that to the averages of your, brain protein and the BSA protein to see if you were able to see more Krebs in your brain samples, and then exactly how much Krebs did you see in your brain sample based on the absorbance and the equation that you get. Okay. And so that information is all listed in here as well. your bar graph in that case is going to be the, looking at the amount of Krebs in your BSA versus your actual grade sample. That's what you're going to be looking for. And there should definitely be a difference. So, any questions? Everyone good? I think it would be useful to bring your computers or something with you so that you can go through the data when we get it at the end. That way I can help you if you're running into any trouble. But again, it's the same idea as what we did with the protein quantification. We are creating a standard curve, and then you're creating quantifying your proteins and your unknowns, and you're using that to create a graph. Okay. Okay. So if there are no more questions, I'm done. I'll stick around to answer any questions about your tech report and your problem set if you have a question about those numbers. And otherwise, I'll see you next week.